So we've seen how the rules for genetic outcomes can be useful for figuring out the genotypes of individuals in a test cross. And something that's probably most familiar to you, we're finally getting to, is being able to predict the outcome of a cross based on known genotypes of the parents. So a very nice feature of genetics is that if you understand how the alleles are inherited and how the alleles contribute to the phenotype, then you can make a really strong mathematical prediction about the outcome of the offspring. So for any one offspring, you could predict the chances that the offspring will have a certain phenotype, the chances it will have a certain genotype, or if you take a pool of offspring, you can take an estimate of how many of those should have one phenotype or another. So we'll work through an example here. We'll do plenty of these as the course goes on. For many students, this is probably the most memorable part of their genetics experience so far. And hopefully you had fun with that, or if you didn't before, you will this time around. I hope to make it interesting for you. All right, so let's say we take a cross of these individuals. We have a dwarf plant on the left and a tall plant on the right, and the tall plant is heterozygous. So we can anticipate the kinds of gametes that these individuals can make, right? So if the dwarf individual has only little t alleles, then it's going to generate gametes that have little t alleles. So the dwarf parent is diploid, has 2 copies of those little t alleles, but any one of its gametes, and in this case we'll make it the female parent, any one of those egg cells would contain just one of those little t, right? And then the other gametes, the sperm cells, would have to come from a different parent in this case. So those are coming from the tall pea plant, and that's where it gets a little more variable, right? So we have a tall pea plant that is heterozygous, and as we've seen before, law of segregation of alleles, if you have a heterozygous genotype, then drawing those gametes from that genotype, each gamete gets one or the other of those alleles in equal frequency, more or less. So I've gone ahead and made it exactly equal frequencies. If you count up the 8 hypothetical gametes here, we've got 1, 2, 3, 4 with big T, 1, 2, 3, 4 with little t. This doesn't have to happen exactly like this, but if you take enough gametes, the average will be basically that there's half big T and half little t. So that's another part of this mathematical prediction we can make for these genetic crosses. So in producing one of their offspring, we're basically doing like you would sort of blind yourself and take one from this group of possible egg cells and one from these possible sperm cells and combine those together. That would be one of the possible offspring. And that happens every time. That's really just an example of the biological process of sperm cells finding egg cells. It's a random process. The cells have equal chance of finding each other, and their genetic makeup is really independent of that. So every egg cell should have a little t, and half of the sperm cells should have a little t. So let's see what we get. So this is just one hypothetical. Imagine I drew one egg cell from the left and one sperm cell from the right. One possibility is that we got a little t and a little t. So what was that chance? Well, it was a 100% chance that we got the little t egg cell, and half of these sperm cells were little t. So 50% chance over here and 100% chance here. Combine those together, that's overall just half of all possible outcomes would get us this little t cell, the zygote that would develop into the next generation pea plant, and that of course would have the dwarf phenotype. So let's do a really standard kind of cross here to illustrate some of the principles we've learned. This is a kind of cross known as a monohybrid cross, and I'll explain maybe how that works out in a little bit. Has to do with this idea of hybrid, which is another way of looking at heterozygous individuals. So in a monohybrid cross, we're going to start with 2 parents that are coming from different genetic lines. So let's say you had 2 plants of the same species and you wanted to compare the genes for making them different in one feature you're interested in. So we'll stick with our tall and dwarf example here. And say you took these plants from different true breeding lines and cross them together. As we now know, of course, that generates your hybrids of the F1 generation. So we can assign, let's say you didn't know anything about tall and dwarf pea plants here, you would figure out, if you were Gregor Mendel, rather quickly, maybe not so quickly, everything looks easy in retrospect, figure out that the gene for tallness is beating out the gene for dwarfness, right? So in the F1 generation, the phenotype of the dominant allele is showing through. And so you can kind of conceptualize that as your two different alleles for this tallness gene, and the taller version, the big T, is going to be the dominant one. So this is kind of where that idea of monohybrid comes into the definition. We have individuals, the mono is we're looking at one gene here. We'll look at a dihybrid example in a second. And so we're crossing two hybrid individuals by each other. The monohybrid concept kind of talks about this whole cross. So sort of working a cross of unknown parents all the way through to the F2 generation. So as we've seen it before, what do we predict would be the possible offspring? We've got a variety of gametes that each parent can make. And I'm lining these up all too orderly to start with. So let's say each of these is a gamete. What are the chances, for example, that we get an individual that is now little t and little t? Well, that requires us to get a little t from the left-hand parent and a little t from the right-hand parent. So we're now combining a half possibility from one parent with a half possibility from the other parent. And there are a few ways to look at this, as we'll see. Maybe I'll preview this for us here. So the most common way people work through these examples is with a Punnett square. And so we look at the kinds of alleles that the left parent can make and the kinds of alleles that the right-hand parent can make. And we put those into combinations. We follow these letters across and down. And we find out that out of 4 possible outcomes for genotype, one of those is little t, little t. So this says there's a 1/4 chance that that would be the genotype of the offspring. And then, as we can see, that's also the only way to have the dwarf phenotype out of these 4 possible genotypes. So 1/4 chance, also referring to the chance that we have a dwarf offspring. And so we have here, this is just illustrating the 3/4 chance that the individual would be tall. So from a phenotype perspective, and phenotype is of course what you'd be able to observe with your eyes, 3/4 of the offspring would be tall in this monohybrid cross, and 1/4 would be dwarf. So this 3/4, 1/4 ratio is something you probably want to get familiar with. Sometimes it's referred to as 3:1, and I'm not as fond of that one because in writing this notation, I think you sometimes forget that it's 3/4 and 1/4. So 3:1 refers to 3 out of 4 and 1 out of 4. So I much prefer to think of this as fractions or decimal ratios or something like that. But a lot of these things, you make sense of them however you can. All right, so our monohybrid cross has kind of this predicted outcome as long as the alleles and genes are playing by the Mendelian rules. All right, let's get this moving. Okay, there we go. So a dihybrid cross, the same thing, but now we're involving alleles for 2 genes. So we have P color, green and yellow, and we have P shape. So wrinkled here and smooth here. So round green was one of our parents, and wrinkled yellow was one of our parents. And we can see in the F1 generation that the offspring are yellow and smooth. So this tells us something about our alleles as to which one is dominant. So let's start, I've got shape first. And so we see that round shows up. All right, round. So we know these P's are round. They also had a parent that was not round. So wrinkled is lowercase r. Round, big R. Again, in these controlled genetic crosses, we're coming in with true breeding parents. Good way to start. And then greenness or yellowness, which one is dominant? The hybrid offspring are yellow and heterozygous. And I'm going to do a funky thing with my Y's here, where I'm going to make them really obviously a little curly Q on there. Depending on your handwriting, just make sure that you can convey capital letters and lowercase letters. This is how I write my Y's, so I just need to make a little curly on the little y to make sure I can tell that it's different from the big Y. So if your uppercase and lowercase look the same except for size, that's something that's easily confusing. Try to keep them from confusing you. All right, let's see here, and forward. So what do we predict for our F2 generation if we have this dihybrid cross where we have two parents that are heterozygous for two genes crossed in the F1 by F1 cross? Well, it gets a little bit trickier, right? It's a little more complicated. You can still do this with a Punnett square, but you'll want to do it with a bigger Punnett square. All right, so let's say we're crossing, put our genotypes up here. We had two heterozygous parents, big R, little r, big Y, getting a little too curly there. And laws of segregation of alleles and independent assortment tell us that we've got possible gametes here, right? So big R can be with big Y, big R can be with little y, little r can be with big Y, little r can be with little y. You can do your gamete square for this one. Two kinds of R, two kinds of Y. And we get the same answer here, keeping our R's ahead of our Y's. R, Y, R, Y. All right, so those are the four possible gametes. Our two parents are genetically the same, so they would both mix in the same way to get these four possible gametes. And that should be the same that's shown in this illustration from your book. So big R, big Y. Maybe I'll put them in the same order as your book has them. Little r, little y, big R, little y, little r, big Y. R, Y, R, Y, R, Y, R, Y. And let's just draw one in there, maybe just jump down to a hypothetical one of these possible, there's 16 possible outcomes here, so I don't have room to write them all. But what I have drawn for this individual is, this is one individual that has the big R phenotype and the little y phenotype for color. So two little y's is green, the recessive phenotype. So you'd be able to figure out the phenotype for these 16 possible genotype of your offspring. And that's what's shown here. All right, looks like we're gonna leave some colors on there. All right, so those are our possibles. In here, we can see there are 1/4 or 4 out of 16 of our possible outcomes are green, and 1/4 or 4 out of 16 of our outcomes are wrinkled. So that shows us actually the ratios of those genes individually are the same, even though we're looking at two of them together. So there's still 1/4 chance of being green and 1/4 chance of being wrinkled. But you can look at these in combination now. So what are the chances of being green and wrinkled. Well, that's 1/16. What are the chances of being yellow and wrinkled? That becomes an interesting phenotype. That's 3 out of 16 here. So this is one way to get at some of these answers. You can draw a Punnett square and total up your possible outcomes. We're going to see some other ways to look at these questions as well. So if you want to look at it without a Punnett square, I think we basically went through these, so you can look at the genes individually and the associated phenotypes, or in this third question, the chances that you get 2 of those phenotypes in combination. So green for the yellow-green gene and round for the round wrinkled gene. You can combine those together. So green and round, 3/16. So maybe we'll keep track of that one. Green and round. What are the chances of that? If you don't want to do a Punnett square, there's another way to look at this, where you take your genes and you look at them individually. So color gene, you can look at yellow. And remember that we had heterozygous parents. Oops, we got an extra curly on there. So this one's a big Y. Sorry about that. Okay, so 1/4 green. If we were just looking at the phenotype for green or yellow, and then we've got our round and wrinkled, that's the R gene. And we know that there's 1/4 chance of being wrinkled, 3/4 chance of being round. So what did we say? We were looking previously at green and round. So we have this branch diagram of 1/4 of the possibilities are green, 3/4 of the possibilities are yellow. And then of those 1/4 green, 3/4 of those are round. So we can combine those 2 together and get this same outcome as we had before. This is gotten by multiplying those 2 outcomes together. We'll do some combined probability a little bit later. So we can actually get to the same answer a little more mathematically. And in this example, we didn't have to do a 4x4 Punnett square. It's a time saver if you're comfortable doing something like this mathematically. If you prefer to 4x4 Punnett square, that'll get you through most of our problems that are like this one. Okay, so as I said, we're going to look more at combined probability, and we'll investigate that next time.